Oh, perfect. Okay. So you guys are going to be doing your first day of notes today. We're going to be looking specifically at the nucleic acids, so the family of molecules that are responsible for producing proteins. But before we get going, yesterday you guys watched a video about DNA, looked at the building blocks of DNA when we built out our little puzzle. So that being said, what are the 4 building blocks or nucleotides of DNA? Okay, and go ahead and put that in the chat. If you don't remember, like I think they mentioned it in the video, the actual names, you may use the abbreviations. So what were those 4 building blocks we use to build a DNA? And hi, Christian, and hi, Nick. So in the chat, what are the 4 types of nucleotides or building blocks used to make a DNA? And please, let's not everybody jump all at one time here. Let's like take turns and, you know, take turns responding in the chat. And feel free to open up your notebook if you need to go back and take a look. That is the type of bond that holds them together. Remember, they were letters. We had 4 different letters that we were using yesterday. There's one of them. So you have A. There we go. A's, T's, C's, G's. Good. We got a T in there. Awesome. Yeah, Kaden has got it in there. So if you were struggling, you're like, I am not sure. That would for sure be a question on your test. So what are the 4 nucleotides or building blocks that make up DNA? That was your A's, your T's, your C's, and your G's. Okay. So to what Lex was saying, those you learned in the video are held together with a special type of bond called a hydrogen bond. And we're going to talk a little bit more about, remember that structure function relationship there, about how the molecule itself, how DNA is put together, and how that allows it to perform the jobs that it needs to for you to stay healthy and alive. Once we go over slide 6, then I'm going to go ahead and walk you through what you guys are doing for your notes today. So our discussion is going to be a little bit shorter. We're just going to correct slide 6. Then I'll get you going, so you're going to have a little bit more independent work today. And then tomorrow we'll go back over our notes and make sure everybody is on the same page. But from yesterday, remember our big goals. So things that you should be able to kind of checklist be able to do. 1, you can define DNA. And you can define what a nucleotide is. Today, we're going to add one more term. So we're adding RNA. So what is RNA? The second thing, you'll be able to model the structure of DNA. And then today, we're going to be modeling the structure of RNA. We're still going to use those models to explain, again, that structure-function relationship for DNA. And then lastly, you guys are going to be able to compare and contrast DNA to RNA. Okay? But if we go back to slide 6, okay, oops, that's slide 5. You guys should have something that looks like this. So I'm going to walk us through each of these answers. So your guys' notebook should look pretty similar to mine. If it doesn't, I would suggest switching some stuff around, okay? But basically, if we look at that first question, DNA contains the code for making what? The past couple of days, I kept saying the code for making you, it's our source of genetic information, all of the instructions for making you. Well, that is absolutely true. However, when we're going to look at throughout the unit, we're going to dial it down a little bit more specific, because the instructions for making you is really the instructions for making all of your proteins. And if you remember back last semester when you guys looked at your biomolecules, so carbohydrates, lipids, and proteins, those proteins, man, they are the workhorse. They do absolutely everything for you. There's not a single process in your body that doesn't have a protein associated with it. So we have proteins that are involved in our building our structure, so muscles, tendons, ligaments, everything to keep your form. We have proteins that are special and specific to managing all of the chemical reactions in your body. We also have ones that are specialized hormones that send messages all throughout your body. So again, these things are incredibly essential for your survival, which kind of makes sense that for a long time, remember, up until the 50s, scientists believed that your genetic code lied with the proteins in your body. While that wasn't so correct, because we know that now it's DNA, they weren't far off, because the DNA really is the instructions for the proteins. It's the instruction manual for how to make them all, okay? So when we talk about the nucleic acids today, and I mentioned, and we're going to do this tomorrow, but we're going to look at it from the perspective of like a family business, like mafia style, the DNA being like the godfather of the nucleic acids. And every proper mob family has got their thing. What did they do? What business are they in? Okay. Your nucleic acids are in the business of proteins. That's their job. They have the instructions and they make the proteins that ultimately keep you functioning. Okay. Now, this next part, when we look at the nucleotides themselves, okay, the nucleotide is the building block of the DNA, okay? Now, when you guys were making those puzzle pieces, we were using them, each nucleotide or building block was one whole piece. That's a little misleading because the nucleotide itself is actually made up of 3 pieces, okay? You have a sugar, this guy right here. And fun fact, whenever you see any sort of like pentagon or hexagon kind of structure like this, that's a dead giveaway that that guy is a sugar, because sugars make what we call a ring structure, okay, which is really, really strong, okay? So you have this sugar piece, and then that is connected to what we call a phosphate group, okay? This phosphate group is made up of different phosphorus atoms. You got some oxygen atoms, and I believe some hydrogen atoms in there. And then attached to that sugar, you have what we call a nitrogen base. And it's this base that's your genetic code. This is the instruction piece. Now, these nucleotides, when they attach, they do something actually pretty cool. So if you remember, I said that the DNA makes a swirl. So it makes this double helix effect. And it has the ability to crunch itself down and squish down to become really, really compact so it can fit in a nucleus. Okay, because we need to store every single cell, minus your red blood cells, have the code for you. They all have a copy. Okay, so that's a lot of instructions. So the DNA has to be able to condense itself down really small, but also be able to stretch itself out, because really we have to be able to read the code. You have to be able to read the instructions. Now, how many of you, I want you guys to think about this like little structure here. These guys right here, your phosphate and your sugar, phosphate, sugar, phosphate, sugar, okay? This is what we call the backbone of DNA. Now this backbone, think like your backbone, not only provides protection, but also support and stability. If we removed your backbone, think about that, you would be just a pile of mush sitting there. You'd be like, well, okay, no form, no structure, nothing. So this backbone, okay, provides that strength to the DNA. Now, these guys right here, this linking effect also does something really cool. So this guy right here, this phosphate group, think of it almost like a ball joint, okay? And you have these sugars that come off of it. So think like this. So if you have a ball joint, think like your hip. You can rotate your leg around, inside your hip, your humerus, your thigh bone, sticks in there, okay, or no, your femur, sorry. Your femur sticks in there and it rotates around, allowing you to move your leg. That's essentially like what these guys are doing. They have these two sugar molecules, one here and one here, that are attached to it and allow this almost like swiveling effect. Remember, DNA is a spiral. So when they swivel, they're able to kind of do this kind of effect. Now that allows them to not only open, think like a hinge, opens, but also for it to squish down and reopen and pivot and switch down. So this guy right here, when we talk about that form function, this guy is really that connecting joiner piece that allows the molecule to have some flexibility. Okay, we can squish it, we can condense it, we can extend it, move it around a little bit. And then the sugar, their job is to hold the code. Okay, so we're going to hold the code right here, and that all three of those pieces together make up one single nucleotide. So you have the phosphate sugar, and we call it a base, so you guys can call it just a base. And then you have a phosphate sugar base, and they're connected right there at those swiveling little joints on the phosphates. Okay. Questions about that part right there with the backbone? And remember, always feel free if I say something that's confusing, go ahead and just use your raise your hand function. I'll stop, I'll pause, or drop your questions in the chat as well, okay? Now, the crazy thing, so that was true for the backbone. Now, the great thing is it's also true because this is what we call a double helix. There are two strands of your DNA. One side, so you have two backbones. Basically, they create a mirror image. And it's these two backbones, think like a book. You have two hard covers on each side that close in the information. That's like what's going on here with your backbones. Okay, you have two of them, squeezes in, and it traps and protects your nitrogen bases, which provide the code. All the instructions lie here. Okay, now when you're looking at the hydrogen bonds or the types of bonds that are used to make up DNA, whenever you see these solid lines right here around the sugar, right here connecting the base, and then connecting the sugars to the phosphates, those are referring to covalent bonds. Now, covalent bonds are much stronger. They're where when two atoms share an electron to form that connection or that bond, and that forms a really strong connection there. Okay, which really makes sense because we don't really want our backbone falling apart, right? So the stronger the connection we can have between all of these pieces, the better. Okay, that's okay, Aiden. Now, when you're talking about the code, though, you want some flexibility because you have to be able to get at the code. We can't have every part of your DNA be on essentially like a lock and key lockdown situation, right? We want to be able to open these pages, look at the code, close it back up. You need to have some sort of way to like hold it together, but also be able to have it exposed. So covalent bonds are not really flexible like that. Once they make that bond, they're pretty much like we're holding on. Hydrogen bonds, however, are pretty loosey-goosey. They're like, cool, I'll make a bond. Oh, I don't want to make a bond. Oh, I'll make a bond. And I'm out. And I'm in and I'm out. And I'm in and I'm out. Okay, think like a zipper. Zoop, 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 zoop. Okay, I like to think of the hydrogen bonds too as like high school romances. Like, oh, we're together. Oh, we're not together. Oh, we're together. Oh, we're not together. That's kind of how hydrogen bonds work because hydrogens are like the social butterfly of the atoms. They make bonds really quickly. It's like making friends, and then they're off making new friends. Okay, kind of how the hydrogens respond in the chemical world. Now, why that's important here is that these hydrogen bonds are basically keeping the two strands together, but they need to be able to come apart so that the code can be read and then come back together again. So this is really when we talk about that form function, the fact that these guys come apart allows the code to be read. So that's the function part. And the structure part is the type of bond used. Okay, so you have the covalent bonds keeping the sides, like basically the sides of a ladder, nice and sturdy, or the ends of a book, the covers, the hard covers of a book. And then you have a more of a flexible bond that allows it to open and spread apart and come back together again. Remember, this side right here allows that molecule to swivel to create that helix or that spiral, and then squish down, come up, squish down, come up, and spin, 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 crunch, crunch, crunch. Okay. Now, based off of the type of nucleotides you're looking at, the A's and the T's, because they tend to be a little bit smaller, they only require 2 hydrogen bonds to connect at those places, while the G's and the C's are a little bit chunkier, so they're going to require 3 hydrogen bonds to connect at those spots. Okay. Lastly, the way to remember which ones that go together, remember these are our base pairing rules we kind of established yesterday, and you guys all have this in common. A's go with T's, G's go with C's, C goes with G, T goes with A, always and forever for DNA. And remember, one way you can think about that is just literally how you would write the 2 letters. A, you use very straight lines. T, you use straight lines, so they go together. G's are curvy, so are C's. So the curvy lines go together, curvy ones go together, straight ones, angular ones go together. Okay, so it's a fun little way to remember that. This, though, is more like what you're going to see. And when you think about how long DNA is or how big it is, this is a model of 2. You're looking at a DNA strand that's going to have roughly 3,600 bases or nucleotides in length. So this thing is crazy. Okay, crazy long. But all of that special info is right here tucked neatly in the middle. Okay. Questions on that part? All right, so today when you guys are going to be learning more about specifically the nucleic acid family, you are going to find that there are some similarities how the other nucleic acids, so think RNA, that's one you're going to learn about today. There are some similarities in how RNA is built, and there are also some differences. So part of your assignment today is that instead of, we're going to skip around in our notebook a little bit. So we just finished slides 4, 5, and 6. Today, we're going to skip all the way down to the explain section to slide number 13. Okay, so using the Nearpod that's assigned to you guys in Google Classroom, you guys are going to take some more solid notes on DNA and RNA, the main family members of the nucleic acids. So big things to make sure you have down to be, okay, what is the function of DNA? Where is DNA found in living cells? Okay, what are some other big important pieces of information about DNA? And you want to really make sure that you're tailoring this information to their job. Their family business is proteins. So try to find things like facts that link it to their role with the proteins. Okay. Now, or RNA, okay, these essentially are like the cousins of DNA. They're the rest of the fam. Now, each RNA, actually, there's a couple different types of RNA. So they serve different roles in the family business of proteins. You have one group that are called mRNA that you're going to learn about. You have one that's called rRNA, and then one that's called tRNA. These little lowercase letters that are in front of their name have something to do with their job. It's kind of having like their little job responsibility or their name tag. Okay, so you guys are going to learn about those 3. Again, you want to make sure you're adding information like what do they do in the process with proteins, where are they found, what are some special things that are unique to each one. I've added some of these. You guys should have those along the side. Remember, those are completely optional. If you find you want to add extra info, you can just drag them over and add it. If you feel like you don't need them, you can delete them too. That's okay. I added one over here on the side as well in case you wanted it for the left side. After you're done taking your notes though, this is where you're going to do just like a quick compare and contrast. So what are some things that are unique to only DNA? What are some things that are unique to only RNA? And then what are some things that they have in common? Because just like a family, we've got different you know, different strokes for different folks within a family. Not everyone's the same in a family, but you do have some similarities. You do have some things in common. And these guys are no different. Try to get at least 3 per column. You can put as many as you'd like there, and when we build it out tomorrow together, we'll add as many as we can, but try to find at least 3 things unique to DNA, 3 things unique to RNA, and 3 things they have in common. Okay? Questions about what you're doing for slide 13? All right. Last thing I want to point out, just so there's no confusion. So in Google Classroom, you guys are working on the lesson 2. You're using that link for the Nearpod. I know sometimes that Nearpods can kind of like bog down your system when it's trying to load a video, and that's kind of irritating. So what I did is I removed the function where it forces you to do every page. So this one you can skip ahead. And usually it's the videos that kind of trip you up. These 2 videos that are posted with the assignment are the videos inside the Nearpod. So if your Nearpod kind of like stalls out and it's having problems loading, stop watching it through the Nearpod, go back to Google Classroom and click on one of the videos, the one that you're watching, watch it that way, and then go back into the Nearpod and just keep going. So you're gonna have 2 videos, they're not very big. One's 1 minute, I think this one's like 4 minutes maybe. And then you have 1 reading section inside your Nearpod. Okay, so this is all the info in case you get stuck in here. So just know this is the same stuff that's in your Nearpod. Okay. Once you are done for the day. So by tomorrow, you just want to make sure that you have slide number 13 completed. You've got slides 5 and 6. Those are the ones we just kind of went over from yesterday and today, so that those are done and be ready to go for tomorrow. And we'll then discuss kind of the similarities and differences between nucleic acids and their roles in producing all of our awesome proteins. Okay. You guys, if you have questions, stick around. If you're feeling good about what we're doing today, you don't need any clarification, you are free to either turn off your mic and your cameras and work in the Google Meet, or you guys can leave. I'll have the Meet open until 1:25. So I'll be here if you guys need me. Otherwise, good luck. Have fun. Have a wonderful day as well. Wish for the sun to come back.